Component 2 test book, question 11. How insulin is produced by the process of genetic engineering. And so here we have a human pancreas cell. So insulin is produced in the pancreas, in your pancreas, and there's our useful gene that is going to code for insulin. And we are cutting that gene out and we're putting it into one of these, which is a plasmid from a bacterium. So remove the plasmid and put the gene in. So we are cutting out, and we're cutting there and then joining the bits together and popping them back into a bacterium. So we only want to talk about this sort of thing here. That's only where our marks are going to come from. So tell us about the cutting. Tell us how it's cut. What we use to cut it. Any more information about any things? Why are we choosing one thing to cut it, and what would we have to do to one and do the same to the other? That sort of thing. At point C, it's usual to insert a gene for antibiotic resistance. So it might have been in the past, but probably increasingly rare to do so now. But why would they insert another gene? Well, these are bacteria, so we can grow bacteria in culture. We can grow them on dishes where they produce colonies. But it's quite hard to see which genes are going to have our insulin gene in. But if we introduce another gene, we can do various tests on it to see whether the useful gene has been taken up or not. And they're using this antibiotic resistance. So think about how would they tell one from the other? How would they know whether what they wanted to go on has happened? What would be the results? Would things live? Would things die? That sort of thing. And then the last part, well, back in the kind of 1970s, when I was a kid, we didn't have this, and instead, if you wanted some insulin, you had to get some kind of pig insulin. And you can imagine the sorts of problems that you might get extracting something from another animal and putting it inside a human. So you must come up with two good reasons. Maybe think of a kind of scientific kind of reason, and maybe an ethical reason there to cover your bases there. And that's the task book.